right, last slide set. Let's go. All right, so what else do we know? All right, so this is going to go into a little more detail about what I told you before. So, while the human body temperature is about 37 degrees Celsius, that's our blood temperature, the inside of our respiratory tract is going to be exposed to air, and not everybody lives in Texas in the summer, so usually that air is going to be a little bit cooler, which means that the human upper respiratory tract tends to be around 33 degrees Celsius, but that temperature is going to vary depending on how hot it is outside. Um, uh, the gut of a bird is about 41 degrees Celsius in comparison. So you got an 8-degree difference there, which is, yeah, pretty significant in terms of the physics of the thing. Everything gets a lot more restricted and solid and more like butter at 33 degrees. And everything's a lot more fluid and in motion at 41 degrees, would be the simplification there. So it turns out that influenza virus proteins have to interact with a lot of different host factors. For example, remember that they interacted with those host factors to get into the nucleus. Do you remember the names of the host factors? Maybe, yeah. You remember karyoferin? I don't know. Yeah, but whatever. You can look it up. It's a couple of these things back. And there are also differences in the interaction partners. So the human nuclear import proteins, the birds will have a version of it, but they will have a different version because birds are not identical to people. Yeah, we have some amino acid differences, even in the proteins. We'll have the same general equipment, but they'll have the bird version, which will just have some differences compared to ours. And so, avian viruses that normally replicate in birds, have to be really good at using the bird version because they have to outcompete all the other avian viruses out there. So, each of these guys is locked in a struggle against other viruses, and they have to be fast and they have to be fit, or else they just get wiped out. All right. So, we also know that avian polymerases, so polymerases from the avian flu viruses, tend to be temperature sensitive. So they don't work very well when the temperature is a little bit cold. So the black circles there are the duck um, polymerase at 32 degrees, and you can just see it's not working as well as the duck polymerase at 37 or the Vic. Those are the normal human virus, and they work real well at 37 because, yeah, that's kind of what we are. So, yeah, they're actually restricted. Okay, so this is going to be two slides, and we'll show you what's going on. So at the top, we've got an assay, like a test that is normally done to show whether or not a virus is present. And so to do the test, we grow a layer of cells. And so the cells are going to be stained purple in all these pictures. And so, like above the avian virus, you see a nice layer of purple cells. That means there's no virus blowing those cells up. That's just purple cells. Those little bits at the corner are just nicks where people put in the virus or where they were. Yeah, these are things that happen during the assay. These are not the actual virus growing. Over on the other side, so rather on the left side, the avian virus is on the right. On the left side, where it says human virus, you see all those white holes. Each of those is a crater. So this is where a virus landed on those cells, and then it blew up the cell it was in after 8 hours. It spread to the cells around it, blew those up after 8 hours, and spread to the cells around that, blew those up, and just keeps making this ever widening crater of destruction. This is called a plaque assay, and it's the same plaque like you would get for your participation trophy, or yeah, the same plaque like you would have on your teeth. PLAQUE, and it just means a like a lesion or like a hole, a spot where something happened. So plaque assay. This is how we count viruses. It would be similar to the way you could count bombs. You could just drop them all and then go back and count the holes and say, oh, yeah, okay, that many bombs. We do this with viruses because viruses themselves are invisible, and because only about 1 out of 100 viruses is actually well enough put together that it will actually start an infection. The rest are all just a little bit genetically broken in some way or have their proteins kind of messed up and they just don't work. Yeah. But that's fine. Viruses have lots of offspring, so it's okay. Okay, second half of this slide. So the crux of this is that you can make mutations in the polymerase basic 2 protein. You can either swap it for the protein from a human, or you can just make a single point mutation, and we'll get to that in the next slide. And that is enough to convert a virus like the one on the right, the avian virus that does not grow in human cells, into one like the viruses in the middle. And look at those gorgeous plaques. You can see this thing grows very nicely in cells, and it actually looks just like the human virus over there. So, this is just another place where the virus can adapt. It can have a particular amino acid to make it good at growing in bird cells, and it has to switch this amino acid to get good at growing in human cells. Because, whatever, you can optimize for any situation. This is the idea. Well, but you can go out there and there's lots of birds. And if you spend a lot of time kissing ducks, I mean, you're going to get your face bit almost certainly. But you would find that there are quite a few of them out there, including some H5N1 avian flu viruses that already contain the E627K mutation in PB2, polymerase basic 2, which remember is one of those three polymerase proteins. You got your PA, your PB1, and your PB2. Yeah, so that's again not great news, right? Okay, so what else do we know? Well, we know that pandemic H1N1 from 2009, so the quote unquote swine flu, although it's like a mix of swine, avian, and human flus, grows very well in mammalian cells at 32 degrees Celsius, which is about the temperature of a human lung, exposed to air that is not the Texas summer. Anyway, yeah, does this mean we would start to catch avian viruses if the temperature gets hot because of global warming? I have no idea, but that's an interesting little speculation. Yeah. I don't know. There may be reasons why it wouldn't work, but yeah, I wonder. I wonder. So the cell actually has ways to detect a viral infection, and we know about some of these ways. So, for example, this is a different pathway, and so what we've got here is TRIM25A, which binds to RIG-I. Oh my gosh, do you remember RIG-I and MDA5? So we're drawing it in a different way, but this is the same thing. See those two card domains? See the big helicase domain? They haven't drawn the little hook at the end, but whatever. And we've got those ubiquitin chains attached to lysine 63, K63. And you can see that it's going over and it's interacting with MAVS, the mitochondrial antiviral signaling protein, and that's interacting with a whole bunch of other proteins here, and these are the ones that we left out of our other explanation, but there they are. We don't have to learn them now, but just so you know, yeah, just so you know where they come in. And anyway, the purpose of this is to activate interferon response factor number 3, IRF3, and also to activate NF kappa B, NF little k b, both of which are going to be transcription factors. So they get phosphorylated, they go to the nucleus, and they will then bind to a promoter and call in a polymerase to make copies of genes like the interferon beta gene. An interferon beta is a message that any cell can send to any other cell, and it will tell that cell, hey, I'm infected and you should start to take action. So you should start an interferon response and start to make all those tetherins and all those trims and all those other proteins. That's the idea. So, yeah, the cell has lots of ways to detect infection, including that same RIG-I system. So, bam, you learned it for Corona, and now you get to use it again. Yeah, isn't it cool how this stuff works? All right. And so, yeah, the first cell gets infected, and if it gets the chance, it's going to make interferon beta, which is going to go over to the second cell, and the second cell is going to make all its interferon stimulated genes. And once that happens, then it's going to block infection of the second cell. And also, it'll start producing interferon beta, and that'll go out to other cells. And so we'll just spread the word that, hey, somebody's infected. And yeah, the more cells get infected, the more interferon beta we send out. And the more we sort of shut down the virus just through the interferon response, which is pretty cool. But of course, the flu virus has got something to say about this, right? There's no way the flu virus is just going to sit there and take that. Here's the flu virus's response. It makes a protein called NS1, which stands for non-structural 1. So remember we had NSP1, that's just what we call the things that aren't incorporated in the virion in coronavirus. This is NS1. And NS1 will go in there, and this is on its own segment. So it's important enough to be encoded on its very own segment as one of the Elite Eight, if you like. And it gets in there and it tries to stop the interferon response. So we know that NS1 will block interferon beta. And so it blocks it at the level of Rig I. And it also blocks it a little bit downstream. So the first place it blocks it, NS1, will actually prevent recognition by Rig I. So it stops Rig I from being able to open up and signal through to MAVs and do all that stuff that it needs to do. It won't get ubiquitinated. It won't lock together. And so even though Rig I has correctly detected that the flu virus is an RNA that has no cap whatsoever, it doesn't even bother with that 2'-methylation. The genome comes in completely uncapped. But the genome comes in with a little bit of NS1 there, and so it will make NS1, and NS1 will shut down the Rig I response as fast as it can. And the other way that this works is that NS1 is going to stop uh, between the point of making interferon beta messenger RNA and the point of expressing interferon beta, which is a little protein from that messenger RNA. NS1 seems to get in at that stage and block it as well. So, whereas coronavirus used several different proteins to just annihilate this pathway at every possible turn, the influenza virus, as far as we know, just uses the one protein called NS1, and NS1 will shut down the intracellular, this component anyway, of the intracellular immune response. So that's kind of neat. All right, and so this is data from Holly Shelton, and Holly Shelton is the one who gave me this talk so that I could use it, so that I could talk to you almost as if I'm an influenza expert, which I'm not. I know coronaviruses, and that's it, and I don't even know everything about coronaviruses. It's too big a field. Um, anyway, so pandemic H1N1 induces moderate levels of interferon. So let's see. The H1N1 is going to be the black bar. So you can see at 6 hours, the black bar is really low. At 8 hours, the black bar is really low. At 24 hours, you get a little bit. And then back at 50 hours, it's low again. Compare that to H5N1. So H5N1, the avian flu, causes pretty good immune response at 8 hours, really strong immune response at 24 hours, a lot of interferons being made, and still a pretty strong response going even at 50 hours when a lot of those cells are dead, but it keeps going. The blue bar is a positive control, so this is just what it looks like when the interferon response is on, and the light blue mock is when everything is off, and the H3N2 this is our other normal human flu strain. As you can see, it doesn't make any interferon response. And the neat thing is, if you were to put interferon in, the interferon does work. It does really inhibit these viruses and also inhibits coronaviruses. But the viruses are so good at stopping an interferon response from being made that it really doesn't matter. They can grow anyway. So pandemic H1N1 is not quite as good at silencing the immune system, but it's trying its best. It has an okay version of the NS1 protein. Whereas your regular H3N2 has a absolute crackerjack version that does a fantastic job. So somebody out there is probably getting infected with H1N1 and might be infected with H3N2. Maybe some unvaccinated goofball that you know. And said goofball, who we can now refer to as Mixing Vessel 1, would be a nice place where you could take the NS1 from H3N2 that shuts down all the business inside the cell and mix it in with the pandemic H1N1, which is a little more dangerous. And bam, we could get an antigenic shift again and get a brand new virus that'll come out and cause horrendous problems. All because, your friend the human petri dish doesn't like vaccines. Yeah, so, okay. Yeah, anyway. These sort of things make me a little bit crazy, but that's not what this class is about. This class is about the science. So I'm going to try and stick back to that. All right. Game face back on. Here we go. Yeah. So, uh, but there are consequences. There are consequences of extremely high interferon induction. So, when you have a lot of interferon response, you can actually overwhelm some of the other cells because interferon signals not only to other cells in the area, but also to cells of the immune system, things like macrophages. And macrophages have all kinds of chemicals inside that they can use to fight chemicals and proteins they can use to fight infections. And when they get too strong a signal from interferon, they just start indiscriminately killing things. They just get mad, in other words. And this can lead to an uncontrolled immune response, a thing called a cytokine storm. And so cytokines are really very similar to chemokines. You remember our CXCR4 and CCR5 chemokine receptors? Well, cytokines are like chemokines. They're like a little protein that can float along and dock onto one of those seven transmembrane receptors. Yeah. And they're also for signaling in the immune system. So there have been cases. There have been cases where you'll get these brand new flu strains and where they will start killing children. And when they do, it seems that it's always because the kids have a really strong immune response and actually make a little bit too much of the interferon, or rather, the virus induces too much. So, in a way, I guess maybe it's better for humanity if we do make that recombinant H1N1 that doesn't induce much interferon, because then we have a better chance of fighting back. It won't cause this nasty side effect. And the H5N1 bird flu definitely causes a really strong cytokine storm, and that seems to be how it kills people. So there you go. I think that's a nice overview of influenza viruses and how they work, and why some are bad and why some are really bad. Yeah. Hope you've enjoyed it, and hope this online lesson thing is working for you. Thanks very much.